We stopped at the last two lessons, ecology. Ecology is, or are lessons 19 and 20. And then we have biotechnology. Okay. Now, the first question that we will go over and revise is the nitrogen cycle. Here you have a figure showing part of the nitrogen cycle. And give me a second. Okay, here it is. Fine. And then you want to describe processes A, B, and C in the figure. Okay, I'm not going to ever stress enough that anytime they give you a figure, you have to refer to the figure. They give you a table, refer to the table. A graph, refer to the graph. So this means that, yes, you will write everything you know about the nitrogen cycle, but don't forget, while you are writing, refer to the figure. So let's go and discuss A. What is A? What is B? And what is C? Let's look at A. You have a dead organism, and then A, and it will be nitrate ions in soil. So what is happening in A? What is A? When we have dead organisms,
Decomposition?
these dead organisms will, whatever inside is the inside the dead organism, it will become nitrate ions. This is
Decomposition.
what. Okay. Okay, I will not tell you it's wrong. The materials or dead organic matter are decomposed, and then what? And
Nitrification?
then what do they release? Excuse me?
Nitrification?
Yes, nitrification. Very good. Okay, now let's look at B. Here I have an arrow from the nitrogen in the atmosphere going straight to the nitrate ions in the soil. So I have nitrogen gas in the air, and then I'm going to trap it or fix it, and it will become nitrate ions in the soil. So what is B?
Nitrogen fixation.
Very good, nitrogen fixation. Now let's look at C. C, I have nitrate ions. They are in the soil, and now they became nitrogen gas in the atmosphere. So what is C?
Denitrification.
Denitrification. Very good. So we will discuss the nitrogen cycle, everything we know about it, and we will refer to the letters, just like we did. Now, we said that we will remember it using NED, if you remember, NEDD. Can someone tell me what is N? What does N stand for?
Nitrifying bacteria?
No, 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 no.
Nitrogen fixation?
Yes,
Nitrogen
nitrogen
fixation?
fixation. Nitrogen fixation. And we will discuss it. Remind me, what is nitrogen fixation? Discuss it and refer nitrogen fixation from the diagram. This is the full answer. What is nitrogen fixation?
Unreactive
In short,
nitrogen gas?
yes. Okay. The nitrogen gas in the air, huh? It will.
It will be converted
Be
to
converted into
nitrate ion.
Very good into nitrate ions in the soil. It's like I'm going to fix it. I'm going to trap it. That's why they called it nitrogen fixation, because the nitrogen gas in the air is going to be directly converted into nitrogen ions in the soil. And who is going to do that?
Nitrogen fixing
fixing
bacteria
bacteria, the lightning.
Yes, nitrogen fixing bacteria. Where are they found?
in the soil or the roots.
Yes, in the soil, either free in the soil or in the roots of some types of plants called leguminous plants. Please study well. Remember the details. I know a student who studied well from a student who did not study well. When the student is confident from the answer and remembers all the details, then he studied well. A student that is hesitant and does not remember the details is a student that studied, yes, but they studied lightly. There's a difference between studying well and studying lightly. When you want to study well and remember all the small details, write it down. Write it down. Okay. When you study lightly, do that after you finish the syllabus and you are studying or revising before the exam. Okay. So this is nitrogen fixation. We said either the nitrogen fixing bacteria are found in the soil, they're free in the soil or attached to the roots of some plants like leguminous plants, or either the nitrogen is going to be fixed by lightning. Someone said it. I heard you. Okay. So I discussed nitrogen fixation and I referred like letter A. No, it wasn't letter A. It was which letter? Letter B. Letter B is nitrogen fixation. So we're done with the N. Now, what is E and D? Because they are discussed together.
Excretion dead organisms?
Yes, excretion or decomposition. Now, this part, I'm very happy that I'm going to revise it. And here I'm going to refer in the diagram with letter A. I have dead organisms here, and they will be converted to nitrate ions in the soil. Now, excretion or decomposition, and I will refer to the letter A. Now, what happens? Pay attention. In excretion or decomposition, any waste products or any dead matter will be decomposed by decomposers, bacteria and fungi. They will be decomposed, they will be digested, they will be broken down, whatever. Use the term decomposed, I prefer it. When they are decomposed, they will release proteins slash amino acids. Now, from these amino acids, what do we extract? What is removed from the amino acid?
Nitrogen part.
Very good. The nitrogen containing part is removed from the amino acid. And the removal of the nitrogen containing part from the amino acid is a process called what?
Deamination.
Excellent. Deamination. Very good. Now, when I remove this nitrogen will then be converted into.
Nitrates.
Ammonium.
No, there is a step before nitrates. Yes, someone said it. Can you say it again, please?
Ammonium compounds.
Correct. Ammonium compounds. And the conversion of nitrates, the conversion of the nitrogen released from the amino acid into ammonium compounds is a process called what?
Nitrification.
No, we still did not reach nitrification. Ammonification. That same question, you can see it with different mark schemes. Sometimes in the mark scheme, they write those keywords, sometimes they don't. So to be on the safe side, we need to know all the keywords. After ammonification, after the making of the ammonia or the ammonium compounds, these ammonia or ammonium compounds are finally converted into active nitrates. This is nitrification done by nitrifying bacteria. So I'm going to revise this part in detail. Again, we are talking about the nitrogen cycle, and our objective is that we want nitrates in the soil because plants cannot absorb anything except active nitrates. Okay? So we will have decomposed matter, decomposition. And we will have waste products. So dead matter and waste products, both of them will be decomposed. Both of them will be broken down. Both of them will be digested. When this happens, the first thing that is released are proteins. They will be broken down into amino acids. And then the amino acids, what do we take from them? Nitrogen. The nitrogen part is removed from the amino acid. The nitrogen that is removed from the amino acid, it is removed from a process called deamination. This nitrogen part that has been removed in deamination is first converted into ammonium compounds in a process called ammonification. And then finally, these ammonium compounds are finally converted into nitrate ions by nitrifying bacteria in a process called nitrification. The nitrogen cycle is very common. And these nitrate ions will be absorbed by the plants and they will become part of the plant in a process called what? When they become part of the body of the plant. Someone give me a name. Are you with me or you're gone? Hello?
Assimilation.
Yes, yes, assimilation. Okay, please stay with me. Now I want to, yes, assimilation. And you can also even add that these nitrate ions will be absorbed by the root hair cells by active transport. So it's not difficult. It's very easy, but just understand it and memorize it in order. I'll say it again for the last time. Anything that will die, okay? Anything that is dead or waste products that will go to the soil. And this question is so common, especially in the recent years. Anything under any organism that will die or any waste products that are released from animals they will be decomposed by bacteria and fungi. They will be decomposed by decomposers. When they are broken down or they are decomposed or they are digested, they release proteins / amino acids. And then what do we remove from these amino acids? The nitrogen containing part in a process called deamination. The nitrogen that is removed is converted first into ammonium compounds in a process called ammonification. And the name speaks for itself. Ammonification is the making of ammonium compounds. Deamination is the removal of the nitrogen containing part. We know this very well. So anything will be decomposed, will give us proteins. We will remove the nitrogen from the protein in deamination. We will convert the nitrogen that has been removed into ammonium compounds. This is ammonification. And then this is the final step. The ammonium compounds are finally converted into active nitrates in the process of nitrification by nitrifying bacteria. And only then will the nitrates be absorbed by the plant by active transport, and they will become part of the body of the plant in a process called assimilation. Okay, this is the nitrogen cycle in full. In full. It is repeated in the recent years. In some mark schemes, they mention all the keywords, the ammonification and the nitrogen fixation and the deamination, everything. And some mark schemes, they don't. But to be on the safe side, especially if this question is out of 6 marks, mention all the keywords and refer to the diagram. In that case, we will refer to A, dead organisms converted into nitrate ions in the soil. And some questions only ask you about A. We solved them together in the past weeks. What happens or the fate of the decomposed matter and how are they converted into nitrate ions in the soil or how are they used up by the plants? Different question formats, same answer. Now, what will you say about C? We said E, excretion, D, decomposition, the last D, the denitrification. What will you tell me about, yes, denitrification?
Nitrogen fixation.
Nitrogen fixation, we finished it. I'm talking about the last letter D, which is represented by C here in this diagram.
Denitrifying bacteria.
Excellent. And what will you say in denitrifying bacteria?
That it takes the nitrogen ions that are still in the soil and converts it back into nitrogen in the air.
Yes, and this is done in soil that is waterlogged or aerated or not properly aerated.
Not properly aerated.
Yes, these bacteria are anaerobic and they are found in waterlogged soil or soil that is not properly aerated, and they are destructive bacteria and they will convert the nitrate ions in the soil back to the nitrogen in the air. That's why farmers keep on plowing their soil so that they decrease the number of these damaging denitrifying bacteria. So here we revise the nitrogen cycle all in one question. It's very important, it's supplement, and it's very common. State the name of the process that plants use to absorb nitrate ions. Now, we already answered this question. What is the name of the process?
Assimilate.
Yes, they absorb nitrate ions by active transport. When they tell you it becomes part of the plant, this is when you write assimilation. So the answer is active transport. Okay. Climate change is one reason why a plant species could become endangered. State other reasons why a plant species could become endangered. Give me any reason why any organism, plant or other, can become endangered. Very easy question.
Deforestation.
Very good. Deforestation. What else?
Pollution.
Pollution. What else?
Diseases,
Soil erosion.
Spread of diseases. Yes. Predation, overhunting. Okay. When you say pollution, at least mention one example. What is one example of pollution that can make
Soil
species
erosion.
become endangered? Soil erosion. Okay. Global warming, climate change, war, whatever. No food, no water. Describe how human actions can cause climate change. Now, this is also a general question. Climate change. What is it? What causes climate change?
Deforestation increases the CO2 level.
Yes, climate change means We have a change in our climate. We have global warming and an average increase in the temperature of our globe. And this results due to several reasons, due to the presence of greenhouse gases in the atmosphere. Give me examples of greenhouse gases. The common
CO2 and
examples that you know.
methane.
CO2, yes. Methane, very good. Now, give me actions done by humans that will increase the level of CO2 in the atmosphere.
Burning of
Burning of
fossil
fossil fuels.
Very good. Combustion, burning of fossil fuels. So this is something that we do every day and we contribute to climate change. What else increases CO2 in the air?
Decomposition.
Deforestation.
respiration.
Deforestation, of course. Yes, respiration. Yes, you're all correct. Deforestation. Of course, less plants, less photosynthesis, less absorption of CO2 from the atmosphere, and more CO2 in the atmosphere. And also more respiration. You are correct. All these factors increase CO2 in the atmosphere, and CO2 is a greenhouse gas. Mention the term, it's a nice keyword, greenhouse, okay, gas that will trap heat and ultraviolet radiation. Don't forget all these details, please. Ultraviolet radiation and will lead to climate change and global warming. Now, what about methane? Methane, when is it released?
Livestock.
Yes, methane is produced due to the or during decomposition, during the incomplete decomposition of organic waste. And this happens from waste products, any type of decomposition, waste products from livestock, yes. Okay. Also, when we pile lots of garbage, remember the landfills? There is some organic waste in the landfills, a pile of garbage from all the houses, and they are broken down. There is incomplete breakdown of these organic wastes in the landfills, and this also releases methane. And the methane is also a greenhouse gas that will trap heat and ultraviolet radiation and will cause climate change and global warming, etc., etc., etc. So we said everything. We said burning of fossil fuels, combustion, deforestation, more respiration, cutting down trees. These gases have a greenhouse effect. They trap ultraviolet radiation or they trap heat. They cause global warming, climate change. Methane gas also is one of the common gases. Okay. These two gases are the most common. You have other gases, but these two are the most common. Now, something like this, a question like this out of 4 marks is very easy. But the problem is that it needs a little bit of memorization. So memorize those details. Now, when you grow one type of crop, this is also a very, very common question. This is called monoculture or polyculture?
Monoculture.
Monoculture. And they always ask you about the advantages and disadvantages, and mainly disadvantages. Describe the negative impacts to an ecosystem of large-scale monoculture. What are the disadvantages? How many? Give me around 3 disadvantages.
No
I'm growing
variation.
one crop. Yes. I'm not growing anything else. So less biodiversity or variation. It has the same meaning. What else? When I have less biodiversity,
Spread
Soil
of disease.
disease and
I did not hear you, but someone said soil.
Spread of
Will
disease.
the soil be improved or it will affect the soil?
unfertile.
Yes, this is going to affect soil fertility. So the soil fertility will decrease because when I grow only one type of crop, I'm not going to make my soil fertile. When I grow many crops, I will make my soil very fertile. When my soil fertility decreases, then I will have to use what to compensate for this decrease in fertility?
Antibiotics.
Fertilizers.
Habibti, I'm not talking about animals. I don't use antibiotics with plants. I'm going to use lots of fertilizers, yes. And these fertilizers, too much of the fertilizers can cause what?
Eutrophication.
Erosion.
Yes, it can be leached out into nearby rivers and cause eutrophication. Okay. Also, what else? More than we said
Spread of
3, but
disease.
what other disadvantage? Yes, my dear?
Spread of disease.
Yes, of course, there's a very high chance that disease will spread. And if the disease spreads and I only have one crop, what can this lead to?
Extinction.
All the
Yes,
crops die.
all the plants will die, and this is going to really hurt my pocket. It's going to affect my profits, and these plants, they will all die. Okay. What else? When I don't grow except one type of plant, but other animals or other insects, they feed on different types of plants. How is this going to affect those insects that feed on different types of plants? Isn't this going to affect the food web?
Yes,
Are you with me?
the
Okay. Huh? Of course, when I grow only one plant, let's say I have an insect that doesn't eat this plant, it eats cucumbers, and you are growing tomato, tomato, tomato. This insect will die. Okay, so we did say loss of biodiversity and variation. We can say that we have decrease in food supplies, and this is going to affect animal populations or insect populations, and it is going to destroy or damage food chains. So I think we mentioned all the disadvantages of monocultures. If they ask you for advantages, what are the advantages of monoculture? In case they ask you for an
no
advantage, why?
competition.
Huh? Excuse me?
No competition.
No competition. You mean competition between different plant species? Okay. But what is the main advantage of growing one type of crop?
Higher yield
Is
of
it?
crops.
Yes. Okay. You are very specialized in growing one type of crop. You will become very good and experienced at this type of crop. It is cheaper, of course, and you will grow lots of this type of
Easier
crop. It's
mechanical
very,
and harvesting.
it's easier to maintain, of course, to grow one thing than to grow several things. So please revise the monoculture and the polyculture. Remind me, what is the polyculture?
Growing more than one type of crop.
Yes. Okay, now this question is repeated. We already answered it. I wanted to show you that they can get the same question in a different format, but the answer is exactly the same. Nitrogen is an important element for organisms in a livestock farm. Waste from animals contain protein. This waste is often spread on farmland as a fertilizer. Describe how the nitrogen in a protein is recycled in the soil into into a form that plants can absorb and use. Now the question looks very complicated, and I have a lot of students that sent me this question. What is this? Is this cancelled? Did we take it? My dear, this is the nitrogen cycle. It is exactly what we said. It is exactly what we said. Who will repeat? I'm not going to say it. Mhm. What happens when
We're,
waste products are released from the livestock?
proteins are going to be released into the soil. Then by
Yes,
deamination,
proteins.
we're going to separate the
Excellent.
nitrogen containing part, and then it's
Excellent.
going to be turned into ammonium compounds
Yes.
using
Very good.
ammonification. The process is ammonification. And then
Very good.
it's going to be turned into nitrate ions using denitrifying
Huh?
nitrogen fixing bacteria.
Excellent. In the
It's going to
process
turn into
of,
uh, nitrification.
yes, nitrate ions. So exactly the same answer. And if you go and look at the mark scheme, you'll find the same answer. Okay, I want to look at the mark scheme because I want to show you that all these words, especially the words deamination and ammonification and nitrification, those are the keywords. Now, I did send you the mark scheme. 1 minute. Let me open it. This is which page? Okay, good. So I want to show you these keywords because you always insist that I don't have to write all that. No, you do have to write all that. Page 51. Where is 51? Here. Let's see. Let's look for the keywords. No, it's here. Yeah, okay. Decomposed, ammonification. It's right there. Converted into ammonium ions, but ammonium ions converted to nitrate ions. They will convert to nitrification, nitrifying bacteria, and deamination. Yes, deamination of amino acids. Okay, the mark scheme throws the keywords, but they don't put it in order. It's your job to put it in order in a nice, decent sentence that are linked. Don't write it like this. Okay, this is just an outline for the examiner to make sure that all these points are included. But you will not write an answer like that, not in bullet points, not in uncoherent dots or points that are not related. Decent sentences in order, they're related and they're easy to understand. Good. So we're done with this question and all its formats. Waste from livestock also pollutes water. Now, of course, here eutrophication is very important and very common. Explain how the pollution of water causes by animal waste leads to a reduction in biodiversity. What do they want to talk about? 6 marks. What's the first word you will mention here? Eutrophication. And mention everything related to eutrophication. The first word you will start with is eutrophication. And describe it. Can someone describe it for me? I forgot. No, someone remind me. You have the exam tomorrow, by the way. I finished my IGs. I don't have an exam tomorrow. You do.
Algae bed grows over the lake.
1 minute. 1 minute. 1 minute.
Reduction
In order.
in levels of O2.
No. Before that,
Can I say
the waste products
fertilizer?
contains what? I want the story from the beginning.
Nitrates and
Nitrate ion.
Yes.
phosphorus.
Yes, yes. The waste products contain nitrates. What you said, both of you, is correct, but I just like things in order. Waste product contain nitrates. Nitrates is food for what?
Algae.
Yes. Please, nice and in order and clear and simple, as if you're writing this question to a child. You want this child to understand you. This algae will overgrow. And then,
Cover the entire lake, a bed of algae.
yes, it's going to cause a green curtain and cover the lake. Yes, very good. And then,
Cover
Blocks
sunlight. The sunlight.
yes, blocks the sunlight. And then, what
The
will die?
water plants die.
Plants don't
Yes, the water
photosynthesize,
plants. Very good, they die. And
decreasing the
after
amount of
anything dies,
dissolved oxygen.
perfect, decomposition, decrease oxygen, very important, decrease oxygen concentration. And then what will finally die?
Fish
will
and
die.
aquatic life.
Yes, fish will die. And in the end, this nice lake will turn into a meadow full of diseases and bacteria, and it's not going to be a nice lake anymore. Now, again, I want to look at the answers for a particular reason. Okay. The first word is eutrophication, and then they start everything here we mentioned, but it's not in order. But everything here we mentioned. They said increased growth of plants. Okay, or algae. We said that. The plants or the producers will compete with each other for light, and the plants or the producers, they will die, or the plants or the producers are decomposed, increase in decomposers, bacteria decomposers, again, they will dissolve the oxygen, decrease in concentration of oxygen, we said it, reduction in biodiversity, death of the fish. Okay, this all are points. You don't write the answer this way. You write the answer the way we wrote it together, in order, nice, clear, for the examiner to understand that and knows that you know the information very well. Okay, this is fishing also. Large scale fishing, this is over harvesting of fish, and small scale fishing. Discuss how governments can regulate fishing to maintain fish stocks. This is a very good question because it includes the regulation of fish, and also use the word sustainable in your answer. Again, 6 marks. Ecology questions are usually 6 marks. How can you regulate fish stock? And then tell me, what does the word sustainable mean? How do you regulate fish? I don't want the fish to finish fishing.
Using nets with large holes for young.
Very good. So use nets with hole size or mesh size that are big enough for who to escape?
Young fish.
Young fish.
Yes, for the young fish to escape. What else?
Protected
Um,
areas.
During
I can't hear
the
you.
reproductive season.
Yes, only
Protected
there
areas.
is something called the closed season. The government is going to tell the fishermen that you are allowed to fish only in a certain time of the year, so that I give time for the remaining fish to reproduce. What else? It's called closed season.
Quotas.
Very good. Fishing quotas. I cannot write properly here. You are only allowed to fish 100 kilograms of fish, not more. What else?
Educating
Protected
fishermen.
areas.
I can't hear you. I'm sorry.
Educating fishermen.
Very good. Education. Now, what we are doing now is that we are educating each other that we have to limit fishing so that the fish population doesn't finish. What else?
Having
I
protected
can
areas
marry
for
it.
fish.
Yes, protected areas, like protecting areas or the fish. We said it. Okay, protecting areas, yes. Protecting certain fishing areas, because these areas, they have very rare fish and I don't want you to come those to fish. I can make fish nurseries. Okay, now these are the methods or the steps that I will do, that I'm going to protect the fish. And I can also make laws and regulations, conservation laws. If you don't listen to my rules and regulations, you will pay a fine, you will go to prison. So these are conservation laws or rules and regulations. Okay, so we wrote everything we can think of to protect the fish. Now we want to use the word sustainable in your answer. What does the word sustainable mean? What is the definition? Who will remind me? Tell me first. Sustainable is a resource that is renewable or non-renewable?
Renewable.
It's a renewable resource. So it's a resource that will be replaced or it will not be replaced.
Can be replaced.
It can be replaced. It is produced as rapidly as it is removed. Now, it can be replaced, yes. But what am I doing here is that I am sustaining it. When I set rules and regulations so that you don't overfish, then I am sustaining my fish population. I am making sure that there is enough fish for everyone to eat and there is enough fish for the future generations. I'm not going to make the fish go extinct, because if I do overfishing without any rules and regulations, I can make whole species of fish become extinct. So when I use the word sustainable, I'm going to define it. A sustainable resource is one that is produced as rapidly as it is removed. It is renewable. Okay, so it doesn't run out. And when I make the fishing sustainable, then I am going to provide for the needs of a human population without harming the environment. I'm going to provide the needs for the current human population, and I will put in mind that I have to also provide the needs for the future human population. So I'm going to do all this so that I keep my fish population sustained. Sustained means maintained. I'm going to conserve it. I'm going to maintain it. I'm going to keep it. And the word sustainable also is very common, and it just needs to be memorized. Go back to your notes and measure and memorize what is sustainable and what is a sustainable development. A sustainable development is a development or a government that is concerned about the current resources, and they are using the current resources they have wisely so that it doesn't finish. And I put in mind that I will still have those resources in the future. I'm putting in mind that there is an increasing population and I have to reserve some of the resources I have for the future generations. Are you with me? Okay.
Yes.
So I'm
Yes.
going to do all this so that my fish doesn't fish. I have my Red Sea or I have the River Nile. If I let anyone fish as they want, the River Nile will have no fish. No, I am the government. I will set rules. You will fish only in June, and each fisherman will only fish 10 kilograms, not more. And you will not fish here because I have rare fish. If I did not do that, the fish population will not be sustained. It will finish. I will not have any more fish for me to eat or for my children to eat in the future. So this is what a sustainable development means. It's a development that will put in mind an increasing human population. Okay, I'm going to provide for the human population now. I'm going to provide for the increasing human population, but I will not harm the environment. I will make sure that everything I have is being sustained. I hope this is clear. Please go back and measure this and study this part, because this part is common. Okay, endangered species also are very, very common. What are endangered species? What is the cause for them to become endangered? And how do we protect them? Now, suggest conservation methods that are used to increase the number of the Arabian oryx between 1978 and 2000. Now, remind me of all the conservation methods that you can think of. What can you do to prevent the extinction of any animal, the Arabian oryx or a rhinoceros or anything? What can you do?
Captive breeding.
Yes, protective breeding. Yes. What else?
Protect their natural habitats.
Yes, or captive breeding. Yes, no deforestation. Do not break down their habitats. Don't break down their habitats. What else?
Stop illegal hunting.
Yes, prevent overhunting. Don't hunt the rhinoceros. Don't hunt the elephants. Also, we can take their sperms and do what?
Sperm banks.
Very good, sperm banks. Also, I can take their sperms and do what? Artificial insemination, in vitro fertilization. I don't need to explain them here, just mention them. Okay? I have only two of those Arabian orcs on Earth, a male and a female. I want children, but they're too old to give birth or they don't do it naturally. So I can do artificial insemination and in vitro fertilization for endangered species to try to increase their population and prevent their extinction. So all these are methods that can be applied on any animal and in plants also, but you will not say sperm banks. What will you say instead of sperm banks when I talk about plants?
Pollen.
Seed banks.
Seed banks, yes. And I don't mention, of course, artificial insemination and in vitro fertilization in plants. Of course, this is only done in animals. Explain the risk to the northern white rhinoceros species as a result of its population size. Now here, explain what are the risks to any decrease in population size. Any animal with a decrease in very little population, what are the risks?
Little variation.
Yes. Now, number one, they can become extinct. And this is something that we don't want. And there's a decrease in their variation and a decrease in biodiversity. And I only have four of this species, four rhinoceros, two males and two females, and they're brothers and sisters. What will happen here? They're siblings.
Inbreeding.
My God. Yes, in breeding, in breeding, in breeding, in breeding. When I have a decrease in population size, I have a problem. They are going to be extinct. And they are siblings. I only have four rhinos, two males and two females. I don't have any other choice except to make them mate together. And this is called inbreeding. Inbreeding means when siblings mate together. Of course, this happens in animals, not in humans. Now, what are the drawbacks of inbreeding? The same drawbacks as self-pollination, if you remember. What are
Disease.
the
Mutation.
drawbacks of inbreeding? Excuse me, my dear?
Mutation.
Mutation? No.
Diseases are more likely to spread.
Yes, yes. Okay, in breeding, I have less biodiversity, less variation. We said it. There is a high chance for the spread of genetic diseases that result due to mutation. Fine. Okay. Now, when they have a lot of genetic diseases and we have inbreeding and less biodiversity and less variation, are they adapted to changes in environment? Are they adapted or they're not adapted?
Not adapted. Not adapted.
Not adapted to changes in environment. Okay, not adapted to changes in environment. They're too old to breathe. We have very little mates. They can't find each other. I want to open the notes and I want to tell you exactly where is it. And so you go back and study it because this is really, really common. Where are the notes? Just a second. Okay. Fine. This is which lesson? I think it's human influences. Yes. Look. Lesson 20, the lesson before the last. It's very important. Eutrophication and all the steps. Eutrophication is very common. And then how forests can be conserved, how plants can be conserved is here. Okay, we will replant plants and we will put plant nurseries and we will educate and protected areas and quotas, education, protected areas, quotas, and replanted. This is copy pasted from your syllabus. How fish are conserved. Please memorize these 7 points or 8 points. It's very common. The reasons for conservation programs. Why do I conserve program. Why do I do conservation programs? To prevent a decrease in biodiversity, to prevent extinction of plants and animals. So I don't damage my food chains. So I protect my plant genes. So I protect my food supplies. And these plants can be used for medicine. So I protect my drug fuels and genes to protect vulnerable ecosystems. You have to memorize it. You have to. 8. Describe the use of artificial insemination and in vitro fertilization in captive breeding programs. Fine. We have to, or we can use these methods to conserve any plants, any animals, sorry, that are endangered. I don't use this with plants, of course. 9, here. Explain the risks to a species if its population size decreases. This is a direct question. This is the question we were just answering. Here are the reasons for the endangered species. We just mentioned them. Here, look at the question. Where is the question? Okay. Suggest conservation methods. Where's the question? Here was the fishing one. And here, suggest conservation methods. I just showed you where the conservation methods are. Where are the conservation methods? Here, conservation programs. Here are the reasons for conservation programs and methods to conserve fish and methods to conserve plants. Okay. And here, the conservation methods for any animals, education programs, captive breeding, legal protection of species and their habitats. And this is the last question we were answering. The risk to a particular species as a result of a decrease in its population size. Those 6 points are so common. I nearly saw them in exams 22, 23, breeding between close relatives, reduction in genetic variation and biodiversity, increase the risk of genetic disease, mutation, more vulnerable to the spread of disease, reduced ability to survive changes in the environment. They are reduced fitness. They're not adapted. They have a very high risk of extinction. Only a small population present, difficult to locate each other, can't find a mate, too old to breed. All this, I did not make it up. It's copy pasted from the mark scheme. Okay. So this is a very common question. Let's finish. Describe how eutrophication of streams and rivers can lead to the death of fish. I'm not going to answer this question because we already did. Okay. I think I went over the common questions in ecology, but this does not mean that you do not study the rest. Study everything. Okay. Genetic modification. Before I move on, does anyone have any questions? Are you with me? Hello?
Yes.
Okay. Any questions? This is the last lesson. We have here steps involving genetic engineering. You will tell me, what is A?
Bacteria.
No, this is the bacterial cell. This is a structure inside the bacterial cell. What are we getting out of the bacteria?
The
And we
protein
will
code.
cut them.
The plasmid.
No, the plasmid. The exam is tomorrow. The protein code is found in bacteria. Where is the protein code found?
Viruses.
In the virus. Okay. Now this is a part of the DNA. Let's say I'm taking part of a DNA from a human because I want to make the insulin gene. What is B? I just gave you the answer.
Human gene.
Yes, very good. Thank you so much. Now, and then this is the plasmid. What did I do to it?
Restricted enzyme.
Restricted
a part of
enzyme.
the plasmid.
Very good. I'm going to cut it with a specific restriction enzyme, and it's going to cut it with what?
With sticky ends.
Very good. Sticky ends. What are sticky ends?
Unpaired chromosome.
Yes, these are short sections of exposed bases. Very good. And then I took the gene out. And then what happened here? What did I do with this plasmid that I have cut and the gene? Excuse me, can you put up your voice?
DNA ligase.
Very good. And what will this DNA ligase do?
To recombine them.
Stick.
Yes, they will fuse them together to create C. What is the name given to C?
Recombinant.
Recombinant.
Very good. Recombinant plasmid. So we just here revise the steps of genetic engineering. We will get one bacteria and we will get the gene from the DNA that we want. From the bacteria, I'm going to extract the plasmid. I'm going to cut the plasmid open with an enzyme. What's the name of the enzyme? Restriction. Wake up with me. Wake up. Your exam is tomorrow. Sleep after the exam. We will cut the plasmid with a specific restriction enzyme, and we will create sticky ends. And then we will isolate the gene. Someone tell me, how do we remove the gene from the DNA? Here it's a plant DNA. How do I remove the needed gene? What do I use?
The same restriction
Thank you so much. Yes, the same restriction enzyme. Why do I use the same restriction enzyme?
to have
To have complementary sticky ends.
Complementary
Yes,
sticky
thank you so much. So to create complementary sticky ends. Now, I'm sure we answered these questions while we were solving classified. All these keywords are important. When you have a question concerning the removal of one gene, putting it in another organism, write this list down, this story, with all the keywords, please. And the making of recombinant plasmid. After we make the recombinant plasmid, what will happen? What is happening here?
The ligase enzyme will recombine.
Yes. And then what? I created the recombinant plasmid. This recombinant plasmid, where does it go after that?
And the crop plant
Yes,
that
it will
needs to
go.
be modified.
No. 1 minute. This recombinant plasmid will go to again back to another bacteria. Yes. And then this
Yes.
bacteria will. This bacteria will. What do we do with this bacteria that has the recombinant plasmid? We only have one type of one number of this bacteria. Is one enough? Someone give me an answer.
Can I make it reproduce and multiply?
Yes,
in a
of course.
fermenter?
I'm going to take this bacteria that now has the recombinant plasmid, and I want it to multiply and multiply and multiply. It will reproduce in a machine. What is the name of this machine?
Fermenter?
Yes, fermenter. Yes, fermenter. Yes, fermenter. Be sure of your answer. So, quickly, the sequence of events. I have a bacteria, the events of genetic engineering. It has a plasmid, and I have the human gene, okay, in a DNA. I'm going to extract my plasmid and I'm going to cut it open with a particular restriction enzyme. So this plasmid is going to be open with sticky ends. I will cut it with a particular restriction enzyme to create sticky ends. And from the human DNA, I don't want the whole DNA. I'm going to only isolate the gene that I need. And I'm also going to isolate the gene with the same restriction enzyme so that I create complementary sticky ends, just like you said. And then I'm going to fuse my plasmid with my isolated gene so that I have a recombinant plasmid. And who will fuse the two parts together? You said it, the DNA ligase enzyme. Now here I created the recombinant plasmid. This recombinant plasmid has to go back to a bacteria. So I will take my recombinant plasmid and put it back in another bacteria. Let's draw it. This is my bacteria. And now it contains the recombinant plasmid. That is part bacteria, another bacteria, and part human. This new bacteria is called the transgenic bacteria because it received genes from another organism. I have to grow it. I want lots of this bacteria. I will put it in this machine, which is the fermenter. And this is a good chance to revise the fermenter. This fermenter, remind me, what do I do to this fermenter to make the bacteria happy and the bacteria will multiply?
Provide suitable temperature and pH.
Very good. I'm going to provide suitable temperature. Here I am monitoring, these are probes, and suitable pH. What else?
Give it glucose.
Yes, very good. I can add a food source, glucose. It will need this in respiration. What else? What does it need to make proteins?
Nitrogen.
Yes, a source of nitrogen. Good. And what will I do so that I prevent overheating of the fermenter?
Have a water
Pull it
coat.
from the
Very good. I heard you. So I'm going to create a water coolant or a water jacket so that it doesn't overheat. Good. And then after the bacteria multiplies and multiplies and multiplies, I don't know what's happening. I'm going to extract the product. I'm going to purify it. And I will take the gene that I want and I'm going to use it in mass production, put it in medicine, in plants, whatever. Very good. Now, this is also a good chance for us to revise the population of the bacteria inside the fermenter and how this population change with time. So remind me of the population curve in the bacteria. We have the population curve of the bacteria with time on the x-axis and the population of the bacteria on the y-axis. How does the curve look like? And what is the name of the curve? And what are the different parts of the curve? What's the name of the curve? Let's start with the name of the curve. It starts with the letter S.
Sigmoid.
Yes, thank you. It is called the sigmoid curve, and it looks like this. Okay, what is this phase called?
The lag
Lag,
phase,
lag phase.
Correct. What is happening in the lag phase?
increasing the population of
Adapting.
bacteria.
Adapting to the environment.
Yes, there is a slow or a fast increase in population.
Slow.
Slow.
Slow. And I heard you saying because it is adapting to its surrounding. Give me another reason why the population is increasing slowly.
It still
They're not
hasn't matured.
Very good. This organism, whether bacteria or anything else, it still did not reach reproductive age. It did not mature. Okay, here. What is this phase called?
The log
Log
phase.
phase.
Very good. It's called the log phase or the exponential. If the log is easier, fine. Now, what is happening in the log phase?
Increasing rapidly of the population.
Very good. There is rapid increase in population. Why?
No limiting
Reproduction.
factor.
Excellent. I was waiting for this keyword. No limiting factors. No limiting factors. Everything is available: food, oxygen, space, mating. They're happy and multiplying and multiplying and multiplying. Okay? Very good. Now, what is here?
Stationary.
Yes, here it's stationary or constant. Good. Now, what are equal in this phase?
The death
Birth
rate
and
and
Birth
the
rate,
birth
death
rate.
rate,
Yes, the death is equal to the birth rate. Good. Now, what is happening here?
death
That
phase.
phase.
This is death. I have now lots of limiting factors that will limit their growth. What are the limiting factors?
Lack of food.
Yes, lack of
High
food,
pH
lack of
levels.
excellent
Breeding height.
accumulation of waste products. It became very acidic, lack of oxygen, lack of food, lack of space, lack of mating, lack of whatever. This is a very easy graph and it's very common. Okay, very good. Now, we revised a lot of things and we did not even answer the question. The bacterial part labeled A is what? What is A? What did we say A was?
Plasmid.
Yes, it's the plasmid. Part A is extracted and cut using a what enzyme?
Restriction
Restriction.
enzyme.
Yes, restriction enzyme. Forming what?
Sticky end.
Sticky ends.
Yes. The part labeled B is the,
Gene.
what is B? Yes, it is the gene. Well done. That codes for the production of beta carotene in this case. The enzyme used to cut part A is also used to cut part B. Part B is inserted into the part labeled A using which enzyme? You all said it.
Ligase.
Well
Ligase
done.
enzyme.
Ligase enzyme. This forms the part labeled C, which is called the what?
Recombinant and plasmid.
Excellent. Yes.
Recombinant.
So this shows you that you have to know the names of the enzymes and the names of the recombinant. The tissue culture is a form of which type of reproduction?
Sexual.
Asexual.
Excellent. Asexual reproduction that is used to produce many identical rice plants, etc., etc. Apart from the structural features, state two reasons why bacteria are useful for genetic modification.
Lack of ethical concern.
Fed reproduction.
Yes,
And
you're both
easy
correct.
to
One of you
store.
said lack of ethical concerns. What else? Hmm?
Rapid
Sharing
reproduction.
universal code.
Yes, someone said that they have a universal code. Yes, they have a universal code. The base
Can
triplets
we
code for the
complex
same amino
molecules?
acids. It can make complex molecules. Yes. Give me all the reasons that you know of.
It can easy to store.
Yes, easy to store. You mean they reproduce quickly and they're cheap?
Yes, yes.
Okay. And they're cheap. Okay. And what else? Did we miss anything? Yes, they have something very important that is only found in bacteria
Good.
that can be transferred. Excuse me? Yes.
The plasmid.
Can you say, please put up your voice? Yes, they have a plasmid, and this plasmid acts as a vector, like a transport medium. It will transport the gene from one organism to another. Okay, easy to
This is
store.
the
Yes, they
plasmid
don't take.
considered a structural feature?
Yes, it's a feature inside the bacteria. Of course it is. Okay. Discuss the advantages of genetically modified rice plants to produce beta carotene. Now, this is a good chance to exploit asexual reproduction, asexual reproduction. Here, what are the disadvantages of genetically modifying rice plants? Now, here we are discussing the disadvantages of genetic modification in general. What are the disadvantages of genetic modification?
Can be
Mutation.
expensive.
Of course, these rice plants can be expensive. What else?
Side effects.
Of course, they have side effects that can be harmful. Okay. They can be harmful to our health. What else?
Cause allergic reaction?
Yes, of course, they can cause allergic reactions. Well done. What else?
They can create diseases?
Of course, yes, they can accidentally create resistant strains of bacteria. And this, of course, will cause diseases. Now, what does it do to the biodiversity? Increase it
Decreases
or decrease it?
biodiversity.
Yes, anytime we interfere with nature, we decrease biodiversity and decrease variation. Very good. Okay. Oh, we finished? Okay, then we finished. Did we revise the advantages and disadvantages of asexual and sexual reproduction in one of the questions?
Yes, that's question.
We did? Okay. Now, who has any questions of their own?
Mrs. Sickle cell anemia cancelled?
Yes, yes, yes. It's surely canceled. But sometimes they will give you a question. I think there was in one question, it's canceled, surely canceled. They will tell you, or they will show you a red blood cell that looks like this, and they will tell you compare that to the healthy one. So you will simply talk about the features of the healthy red blood cell and how it is different. The red blood cell is biconcave. It has no nucleus. It carries oxygen. It is small enough to enter the capillaries. It can squeeze through. Now, all this is not found in this one because this is the diseased red blood cell. But we took other things last year, or no, the 2 years before. That is all canceled to your advantage. Fine?
Okay, thank you.
Okay. Anyone has any questions? You want me to revise anything? Anything is not clear? Now, of course, I have students sending me, do I study these questions only? Okay, what do you think? You will only
No.
study these, that's it? No, of course not. Like these were just expectations, the most common questions in the past recent years. But what if they don't get any of that and they get something else? So study everything, please. And now I'm sure that studying is finished. What do you do now? Because I'm still receiving lots of what do I do now? What do I do? You read. You take your notes and you sit down in a nice, quiet place and you read. You read or you solve past papers. This is what you do now on the night of the exam. And please sleep early. Do not stress yourself. Do not stay up all night until the time of the exam. You will be too stressed because to focus and to remember the details, you have to sleep well. And don't have a heavy breakfast because when you have a heavy breakfast, you become very sleepy. Have a light breakfast and go to the exam. And inshallah, I have very, very high hopes in you. You will do really well, inshallah.
Miss,
Okay.
what
When
are the
is your
parts that
exam?
are in
What
the new
time?
phase? It's 3 p.m. We
3
start
p.m.
writing at 3 p.m.
Okay. Okay. Very good. Please go 1 hour early. Okay. Anyone has any questions?
Miss, what are
Doctor,
parts that are added newly in the syllabus? Because they usually ask questions in these.
The only part that was added in the syllabus was the reasons for variation, the mutation and the meiosis and the random mating and the random fertilization. You're lucky. The new syllabus, we had things removed, not added, and things changed or moved from the syllabus, from the supplement, it became core, or from core to it became supplement. So the new syllabus was actually smaller than the old syllabus. The old syllabus was much bigger. So you don't have lots of additions, you have lots of removals. The
Okay.
dialysis was removed, the a lot of things were removed. I don't remember what has been removed now. What has been added was the homeostasis or the skin. It was core, now it became supplement. And the reasons for variation, that small part, that's it. And a lot of things have been removed. So the syllabus became smaller, not bigger. Do you understand?
Yes.
Okay. And everything that has been removed, it's not in your notes. So anything in your notes, you saw, you know it, you study it. I want to revise this part of the importance of producers. It's such a simple question, but it's common. Producers, these are the only organisms that do photosynthesis, and they trap the light energy and they convert the light energy into chemical energy. And as we eat, this chemical energy is transferred from one organism to the next. But as the energy is being transferred, 90% of the energy is lost. That is why we cannot have more than 4 or 5 organisms in a food web, because we don't have enough energy to support a 6th organism. This part is important. Is it clear?
Yes.
Okay, go take a short break and then sit and read and study and solve. And if any question stops you, send me the question and I will answer immediately. Okay, and I'm with you even at night. I'm not going to sleep early today. I'm with you. If you have any question that stops you and you need help, send me the question, and when I see it, I'll answer you immediately. Good?
Okay,
Yes.
thank you.
Thank you,
You're
Madam.
Thank you,
welcome.
doctor.
Best of luck tomorrow. Bye bye.